Hey, everybody. It's good to see you. Welcome back to Pearson Plus. My name's Andy. I'm here to help you out with your questions involving evolutionary biology and phylogenetic trees. So let's jump right in. I'm going to do a quick screen share with you with the question and some of my notes, and then we're going to get after it here. All right, you can do this. We can do hard things. So we're going to look at a data table. We're going to talk about some relationships and evolution in between species. We're going to translate some Latin names to some common names. There's a lot going on here, so let's jump right in. You know me, the first thing we're going to do is we're going to read through the problem carefully. We're going to try and identify key components of information that we're going to need to help solve these problems. Excuse me, and we're going to work together to figure out a solution that makes good sense. We can do this. You ready? All right, let's go. All right, let's read the question together. The mitochondrial ATPase proteins were sequenced for 5 different vertebrate species. The following table shows the number of differences in the amino acid sequences between each pair of species. All right, let's slow down here and take a close look. Whenever you're given a data table, slow down and really take a look. Make sure you understand what it's showing you. Okay, so let's look at the title. The number of amino acid differences in ATPase among 5 different species. So on the vertical axis over here, they list 5 different species, and they're given their scientific Latin names, genus and species. Across the horizontal axis, same. All right, and then they've got some numbers which represent the number of amino acid differences in ATPase among these 5 species. Let's slow down and think about ATPase. ATPase is an enzyme. Enzymes are proteins. Proteins are made of amino acids. So slow down, let's back up. Anytime you see a name with ASE tagged at the end, it's almost a surefire bet that that's going to be an enzyme. We've talked about enzymes before. Enzymes, their job is to facilitate chemical reactions in the body. This one is connected to a molecule called ATP, which is essential for cellular function and energy and those kinds of things. Don't necessarily know that what the question is going to ask, that's important information to know, but just being able to wrap your head around, like, this is a protein, it's got a job. Its job is an enzyme. It's here to make a chemical reaction involving ATP go for all of these species that are listed in this data table. Now, these numbers represent the number of amino acid differences. If proteins are a chain, amino acids are the links, the individual links that link together. And the terms that we've used before, a polymer is a long molecule made up of a series of monomers that are bonded together. Well, if you bond a whole bunch of amino acids together, you wind up with a polymer called a protein. This particular protein is an enzyme. This particular enzyme has a name, it's ATPase. So what we're looking at is this molecule, this ATPase enzyme, is in all of these organisms. However, it's not exactly the same. There are links in the chain that are different. So the amino acid differences are what we're measuring here. Oops, sorry about that. Let me undo that highlighting. There we go. So let's take a look at this chart in more detail. I've taken the liberty, if you will, of doing some common names. So thank you to the world of Google. The Latin names, the scientific names, I'm going to translate so that you get an image in your head. And careful, I don't want to trick you with this image, but we're going to look at their shape, how they appear, and then their actual evolutionary connections. So Epheris is a wild horse. D. Polyepsis. Oop, I grabbed the wrong one. Is the black mamba, one of the deadliest snakes in the world. G. Gallus is our red jungle fowl, kind of like a wild chicken. The emperor penguin is A. forsteri. And the African wild ass, which is essentially it's very much like a domesticated donkey, is E. africanus. So now I've put some images in your head. Some of these creatures to me seem very similar, like the African wild ass and a wild horse. They share a lot of properties in common. A lot of characteristics of those two creatures look and function very similarly. Whereas like an emperor penguin and a black mamba, seem very different. So, but what does the DNA say? That's kind of the interesting part is like if DNA is so vital and so important, what does the DNA say? And that's what we're going to examine here. Okay. Now I've taken the liberty of highlighting these zeros for a reason. If you look at they list the 5 species here and the 5 species across, these two, this box is going to be a representation, this highlighted 0 right here is going to be a representation of comparing the DNA of the wild horse to the DNA of the wild horse. Now, seems like they should be the same, and sure enough, no difference is found. When you compare the DNA of the black mamba to the DNA of the black mamba, you get 0. So this diagonal highlighted column represents comparing the species that are same, they're identical. So that makes sense. I'm going to push on here and come back to the data table. Let's take a look at the question. It says, using that data in the above data table, create a phylogenetic tree. Now, phylogenetic tree, you can see they've given us a template at the bottom down here to use. Phylogenetic trees are visual representations of evolutionary history. Where in the evolutionary timeline did species split off from a common ancestor? So when you see these branching points, you see these branching points in this diagram. Branching points in the diagram are showing separation of species. We can have a whole discussion about the definition of a species and what that means, but the differences at that point got so great that there wound up being reproductive isolation. They were no longer able to successfully reproduce, and they started to drift apart as a gene pool. Okay, so those little branching points are really key spots in the phylogenetic tree. I'm going to start over. Using the data in the data table, create a phylogenetic tree by appropriately placing each of the 5 organisms, I think they're referring to these 5 organisms at the top, on the template phylogeny below. That's this template. To accurately display the evolutionary relationship between each species. In other words, we're going to put these species at the tops of these endpoints here, and we're going to sort them out based on the DNA data that we have. Technically not DNA data, I'm sorry. The direct data is amino acid differences, right? So we're looking at the protein. Now, DNA codes for the protein, which links to amino acids, but ultimately what we're actually looking at here is amino acid data, not DNA data. Okay. Then it says, in your own words, explain whether amino acid sequence data or morphological data is most likely to reveal the actual evolutionary relationships in these species and why. I'm going to highlight that point because I'm actually going to answer that first. Okay, and then it says, what key concepts are necessary to understand to solve this problem? Let me address the amino acid versus morphological data. First of all, I think I've shown that amino acid data is the differences in the protein. Okay, what's morphological data? Morphological data is going to be form and function. Morphology is the form of how structures and organisms look and operate. And that can give us a lot of good information. That's a lot. All we had for a really long time was either through living or fossilized evidence, how things looked and what the connections that we can make between their form and their function and similarities between organisms that way. Morphological data isn't as good. It's not as good. When you have amino acid sequences that can, in a very concrete and very real way, can connect living organisms based on biochemistry, it's a much more sound way to make decisions than just based on form and functions. Because we've got some things that we know look really different, yet share quite a bit of DNA in common. Humans and bananas look very different. I mean, I don't think, except for maybe my haircut, don't really think I look like a banana at all. Certainly don't function like a banana. But knowing that like 60% of our genetic material is similar, 26% of our proteins are going to be very similar, that's a much higher number than I ever would have guessed. Also, you can get confused between structures that are truly homologous. And truly homologous means they evolved from a common ancestor. They have the same ancestral tie. And features or functions and characteristics of living things that are analogous, which is they look like they are, but they're not. So analogous structures, a classic example is the wing of a bird and the wing of a bat. Look very similar, function very similar, but they are analogous. They have the same form and function, so their morphology is very similar. But they are not homologous. They don't have the same ties in evolutionary history. So because things can looks can be deceiving, right? Don't judge a book by its cover. Looks can be deceiving. So that's a piece of this, is the looks can be deceiving when it comes to morphology. Now, let's see if we can place these based on the DNA. Sorry, not the DNA evidence. It's when we look at amino acids, it's indirectly referring to the DNA evidence, but we're looking specifically at the protein and the amino acid sequence. All right, let's take a look. Well, as we're analyzing this data, I'm going to start with E. ferris right here, the wild horse. It has a ton of differences between the amino acids in the ATPase for a wild horse and the black mamba. That's a big difference right there. I'm looking at all the other numbers going across. Ooh, this one stands out to me also. This one stands out to me as being really close. Ooh, that sense morphological data, the way a horse and a donkey sort of look, and the DNA, or sorry, the amino acid sequences. I keep saying DNA. It's not DNA. It's amino acid sequences. And amino acids are showing that they're actually pretty similar. Okay, that's interesting to find out. And then it's also showing me that the black mamba has a lot of differences. These two are starting to show up as maybe in-betweeners. Hard to tell. Let me go to the next row. This is our black mamba. So comparing a black mamba to a black mamba, no differences. Quite a few differences between the black mamba and the jungle fowl. Quite a few differences between the black mamba and the emperor penguin. Quite a few differences between the black mamba and the African wild ass. It's like, whoa, okay. These numbers are all big, 18, 15, 16. The larger the difference, here's the general pattern, the general trend. The larger the difference, the more time has been allowed to pass in between their evolutionary histories. So they diverged earlier if that number is larger. Let me say that again. The earlier that they diverge in the phylogenetic tree, the larger that number can become. So when you see big numbers like 19, 16, 15, 18 on this chart, those numbers stand out to me as being significantly larger. You know, those things have split early on in our evolutionary tree. So if we slide down and take a look at our diagram, we're going to have to place these organisms on this chart. So because when I compare the black mamba to all the other creatures, I see the largest number of amino acid differences between them. I'm going to drag that black mamba, and I'm not going to commit myself to it, but I'm going to think the black mamba is the one that diverged right here from this common ancestor the earliest. Now, as I go back up here in my problem-solving mode, I'm looking for my experience in doing phylogenetic trees is the farther apart that the species are on the tree, the easier it is to identify. So my brain has a tendency to want to go, well, what's the one that clearly branched off the earliest? Now, what's the one that branched off the latest or is still traveling on that branch? And because these differences, when you compare things to the wild horse, very few differences, especially with the African wild ass, I am going to move my wild horse and I'm going to put it at the end of the line. To me, that is demonstrating this pattern pretty well. Now, the wild horse and the African wild ass only had one difference. So to me, that is a telltale sign that the divergence here on this phylogenetic tree was very recent, that those two were so closely paired that there was only one difference in their amino acid sequence. All right, so I'm down to my jungle fowl and my emperor penguin. I want to see if it matches all of my patterns. So when I compare my emperor penguin to my jungle fowl, I only see one difference here. Oh, by the way, half of this data table is empty. You could fill it. if you really wanted to, if it helped you see it, it's just going to be a mirror image around that yellow 0 line. It's just the data repeats. So we can leave this blank and it's not going to affect our ability to sort of problem solve. But this 1 is another number that stands out to me as being interesting. It's the chicken compared to the penguin. So it means that those two species split in the evolutionary tree very close to one another. Okay, so I'm going to put them down here. And they're both birds, so that sort of makes sense. So as an initial attempt at sorting these 5 species, my confidence is getting pretty high. I really feel strongly that the black mamba is the earliest divergent. The data shows that. I'm really confident that these 2 are close together, and I'm really confident that these 2 are close together. The one thing I'm not as confident on is how do I know I have the 2 birds here, the jungle fowl and the penguin, in the right place, and the wild ass and the horse in the right place. And it's got to be in the data. There's got to be a pattern there that's identifiable. And I think the one that we're really going to look at is the fact that the differences change as we go along the line. So I'm going to look at the top row here, the E ferris. I see my 1, which really closely connects me to my African wild ass. And then I see the 19, which clearly separates me from the polylepsis, the emperor penguin. Oh, I'm sorry, the black mamba. Now I have these 2 sitting right here in the middle. And I am confident that my placement is correct. The question is, does it matter? Could I do it this way? Could I put the red jungle fowl here, emperor penguin here? Answer is yes. These sort of divergent markers, you can pivot around those and you're still showing the same relationship. And so I like my placement like that. So let's see if we've answered the question, right? We always want to make sure that we've done our due diligence in making sure our response is complete. Did we create a phylogenetic tree? Yeah, we sure did right here. In your own words, did we explain whether amino acid sequence data or morphological data is better? I think I made a pretty good case for my amino acid sequences. And then what key concepts are necessary to understand this and to solve this problem? I think the big concept, there's lots of things along the way, like understanding proteins and understanding enzymes and understanding morphology. But the big concept down here is this idea of the common ancestor. This idea is that at some point in evolutionary history, species diverged. The longer ago that divergence was, the greater the differences will be in their proteins, in their amino acids, ultimately in their DNA, in their genetic material. Those differences start to show up. And we as scientists have the ability to track and measure that in lots of cases now, which help us sort and organize these things into truly accurate evolutionary partnerships. Okay, so I am going to come back to you, and I think I'm going to come back to you. Let's see if I can do it here. We're going to stop the screen sharing. Here we are. I'm back. I hope that helped you understand the nature of a phylogenetic tree and how amino acid and protein structures can be connected to that, and how as scientists, we can put all of those together to form a complete picture and a more accurate picture of evolutionary history. Hey, I had fun working with you today. We're going to keep working together. We're going to get better and better every time. We can do hard things. And if there's anything you need from me, please reach out. I will be happy to get back to you and answer all your questions. Take care. Keep working hard. Be good.